Hello. Today we will talk about the vaccine development for COVID-19. Specifically, we'll talk about the different technology that is being used, where we are within the clinical development space, and what one can expect in the near future. There are several technology platforms that are being used for the development of COVID-19 vaccine. Live vaccine is when you have an attenuated virus that one uses that can cause an immune response, but it does not cause a disease. Examples of live virus are measles, mumps, and rubella. There's a well-developed infrastructure for live vaccines. However, safety has to be really well tested in these live vaccine settings. Inactivated vaccines is when you have either a killed vaccine or an inactive form of vaccine that virus which is being used. This again mounts an immune response but does not cause the disease. Example of these vaccines are hepatitis A, rabies, and flu. These vaccines, inactivated vaccines, are not as strong as the live vaccines, so one might need multiple doses or booster doses to provide long-term immunity. When it comes to genetically engineered vaccines, that could be either a DNA or an RNA, when introduced, can form the protein of interest, and it can mount an immune response when virus gets into the body. There are currently no approved genetically engineered vaccines and for humans. Moving along, there are viral vector vaccines, wherein a gene of interest is introduced into a viral vector. There are two types of viral vector vaccines. Non-replicating, that means these viral vectors do not replicate in the body. One, and second one are replicating, that means these viral vectors replicate in the body. There are no approved non-replicating viral vector vaccines. However, when it comes to replicating viral vaccines, we do have for Ebola and dengue. Protein-based vaccines similarly introduce the protein of interest. There are currently a couple of examples of the protein-based vaccines would be subunit for hepatitis B and shingles. Virus-like particle has what is known as a virus-like particle. We're not exactly introducing the virus, so not likely to cause an infection related to the virus. Example of this is an HPV virus. Lastly, on this is repurposed vaccine. That means these are vaccines that have been used for other diseases, but not being used for COVID-19. Examples of this is the BCG vaccine, which is being used, is just used for prevention in TB. This is being tested in phase 3 studies in Australia. So currently, there's a big landscape. There are about 150 vaccines in development for COVID-19. Majority of them are in the preclinical stage. A little over a dozen are in clinical development. And you can see on the slide quite a few of those which are in clinical trial, but it's not a complete at this point. There are a few which are left out. On the very left, you can see the different candidates. In the second column, you can see where they're being developed and which is the company who's actually running these studies. Depending upon which part of the world you are, you might recognize at least one or more of these, like Moderna and Innovia, starting off from US, but the whole world is taking part, as you can see. So it's a global thing. So US is developing, Europe is working on it, studies happening in Asia, as well as in Australia. In the third column, we talk about the different technology platforms that are being used. Some of these technology platforms are easy to develop in the sense that some of them it's faster to bring them to the clinic, like the genetically engineered ones, might not necessarily be the best one. One would have to wait for the data to say what works. So we have all these different technologies, platforms that are being tested. In the very right column shows the different clinical stages of development that they are at. Phase one is more along the lines for safety, has less than 100 patients. For the the phase three studies can have 20,000 patients, and we go more depending on the infection rate. As you see, there are several different technology platforms that are being used. We do not know which one will work, which one will not. It's quite possible that COVID-19 might have a preference for one or the other. It's also possible that the subpopulation of patients might do well with one versus with other. When we say subpopulation, it could be old people, pregnant women, children, immunocompromised. So all this is all these things need to be understood, and the data will tell what would potentially work. These are on several stages of development. Majority are in preclinical, and they have to pass through different stages to be successful to move to the to get to approval. It's not going to happen that all of them will be approved. A small subset of this will be approved. Realistically speaking, we don't live in an ideal world, so you should not expect 100% infection infection prevention with these vaccines. When it's be realistic, and likely that one could have potentially 60% improvement in the infection rate, which would potentially lead to decrease in hospitalization, the severity of the disease, morbidity, and death. Usually takes about 5 to 10 years for vaccines to develop, but a lot of the timeline is being compromised, and the hope is that this will potentially take about 12 to 18 months. The companies have started to manufacture these vaccines even before they are ready before they're approved. So that as soon as the approval happens, things go directly into people who need it the most. After this, I will be talking about the different drugs in development for COVID-19. That will be my next presentation. So please be sure to like and subscribe. Thank you.